Hi, good afternoon, everybody. Thank you for joining us today for the UIC Global Health Colloquium. This is a monthly lecture series sponsored by the Health Science Colleges. My name is Allison Lofthouse. I'm Assistant Dean for Global Health at the School of Public Health, one of the co-sponsoring units. With us today is Dr. Brian Murphy, and he's here to speak with us about establishing a microbial antibiotic discovery program in Northern Vietnam. Before I get started with his introduction, I'd like to just make a quick plug for our lecture next month. Just to mention that December 1st is World AIDS Day. So we always like to have a colloquium that's kind of around that theme. So we're really excited to have with us the Associate Dean for Global Affairs and Planetary Health from the Yale School of Nursing. His name is Dr. Laurent Nelson, and he'll be speaking to us about social, structural, and behavioral approaches to HIV prevention among gay and bisexual men in Ghana, West Africa. Please be mindful that this colloquium next month on Wednesday, December 7th starts at 12:30. Typically, we do start our colloquium series or our lecture series at noon, but this particular one next month will be at 12:30. So we hope you can join us. All right, so I'm going to briefly introduce Dr. Brian Murphy. He is a professor in the Department of Pharmaceutical Sciences here at UIC. He earned his degrees in chemistry from the University of Massachusetts. That was his MS degree, and his doctoral degree from Virginia Tech. After spending 2 years of postdoctoral training in microbiology and chemistry at the Scripps Institute for Oceanography at UCSD, he was hired by UIC in 2010. His lab focuses on applying high throughput robotics and bioinformatics toward the discovery of antibiotics from aquatic bacteria. His lab also partners with local community centers such as the Boys and Girls Club of Chicago to perform high-end biomedical research. Dr. Murphy's program has been featured in Chemical and Engineering News, National Public Radio, Science Daily, the Toronto Star, and the London Science Museum, among others. So we're really excited to have Dr. Murphy with us today. I want to mention that there will be some times for questions at the end of his talk. Please feel free to raise your hand and we will ask each individual to unmute themselves to ask their question. So thank you very much. Without further ado, Dr. Murphy, please go ahead and get started.
All right, excellent. Let me just set this up. Okay. All right, I assume you can see the full screen now?
Yes, we can.
Excellent, okay. Thanks for the kind introduction, and I'm really looking forward to this talk. I've actually never given a talk on this topic before, so it was kind of exciting to put the slide deck together and actually try and tell this story. It's not the typical presentation that I'm used to giving. I'm usually giving presentations that are kind of really in-depth into the science behind our story, but this is going to be a little bit different. Today's presentation is going to be more of my narrative on how a global partnership formed, how it was maintained over the course of several decades, of which I'm only one of those, and some of the lessons we learned along the way to how to build, maintain, and direct a global partnership, because it's very rewarding, it's awesome, and it can be confusing at times. So if there's any early career or younger scientists in the audience, I encourage you to ask questions. And if you want to get into a research field like this, there is no playbook to how to do this, and everything that I'm about to tell you is going to be specific to this partnership here and does not necessarily apply to others. But there are some common rules that I can maybe help give guidance on. And I am by all means not an expert on this either. I'm learning every single day in this. Okay, so let's get underway. My story is going to be really told in 3 chapters. The first chapter, it's necessary for me to give a background of collaboration between UIC and a few institutes in Northern Vietnam, because the collaboration predates me. I came to UIC in 2010, and the collaboration goes back to about the 1990s, the one that I'm talking about at least. The second part of the talk will focus on my new wing of the collaboration, where we've partnered with some scientists in Northern Vietnam, particularly Pham Van Quang, in the Vietnam Academy of Science and Technology. And I'm gonna tell the story of how we decided to combat or use basic and translational research to combat TB. And it's a story about training young scientists and building capacity development. And finally, I'm going to give kind of a vision of where we see our collaboration going in the next 10 years. So before I get to chapter 1, I feel like because it's such a broad audience, I want to give a little bit of a background on who I am and what my research program does, because I think it's really important to understand that moving forward. I'm not an infectious disease researcher, and so maybe this is going to be a unique seminar in that sense. I am a basic science researcher. My degrees were both in organic chemistry. When I did my postdoc, I studied organic chemistry and really marine microbiology, and I've since branched into freshwater environments. And the main topic that I study is natural products chemistry. And this is my current lovely research group to the right. And what we really focus on is trying to discover antibiotics from bacteria that we collect in the ocean and freshwater environments. To me, it's fascinating. It's always been fascinating. And it allows us to go on a lot of different adventures. And I'm going to just lay the foundation for natural products chemistry in the next couple of slides or so before I get into our collaboration. I've got 2 slides of gratuitous photos from our collection expeditions. So we have the great fortune to go and partner with a lot of different institutions around the world. This is just one slide that shows our collaboration with the University of Iceland. We've been able to go all over that island, either collecting from Icelandic Coast Guard vessels or doing scuba diving. The water is incredibly cold. It's about -1 degree Celsius. Which I think translates to like 30 Fahrenheit or 31 Fahrenheit or something. But that's as low as our gauges went down. It could actually be below that. So we've gone and collected in these environments. And the reason that we do this is because we're looking for microbial diversity. And because there's a long history, almost a century of researchers looking for medicines from microorganisms, we're increasingly have to go to different environments and unique environments to try to find both new microbes and new antibiotics that they produce. And so here are just some awesome photos of things that we've been able to do. And to the right is us swimming between, this is actually the cracks of 2 continents. This is the continental fissure. The Eurasian plate will meet the North Atlantic plate here. Really cool dive that was. But we don't just go to fancy international environments. We do regional and local collections as well. And we're always training students in this process. In the top left, we've collaborated with the Environmental Protection Agency and gone on their Lake Guardian vessel. This is a vessel that goes throughout all the different Great Lakes, and we were able to collect sediment. We have students who are organizing expeditions to local state parks. This is down near Calumet Fisheries, one of my favorite fisheries down in Chicago. We've also led an expedition to the Apostle Islands and Lake Superior. These are beautiful sandstone bluffs with these little freshwater caves. And it was here where we did a lot of shipwreck diving because we were looking for sponges that grow off of the substrate of shipwrecks. Sponges are notorious for harboring really interesting microbial communities. And all these photos to the right were actually taken last week. So we have an awesome grant funded by the Illinois Indiana Sea Grants and NOAA. This is to partner with the James Jordan Boys and Girls Club. And I've always wanted to, I've always had a passion for outreach, but I always thought that, antibiotic discovery was actually a field I thought that middle school kids could do. And I was always prohibited from doing this because it's not safe for kids to work with bacteria, obviously. But we've recently, in the past couple of years, acquired a robot that allows a lot of the front end of antibiotic discovery to be done in an automated fashion. And so now we wrote a grant and we're able to work with middle school youth and introduce them to different STEM topics at the beginning phases of microbial discovery. And they get to learn about careers, but at the same time they're learning about careers and doing experiments. They're actually doing high-end biomedical research. It's a super cool program that's pretty near and dear to me. And they're all trying to collect natural products. And I think it's important for me to probably try and define this. Natural products are synonymous with small molecules or secondary metabolites or specialized metabolites. And my definition is that molecules that really aren't essential for the immediate survival or growth of whatever organism is producing them. And what produces these natural products? Well, basically everything. You have microbes or plants or even both marine and terrestrial organisms like sponges, phytoplankton, etc. And to give you an idea of the size of what we're talking about, I think a lot of people in this audience are familiar with proteins. And so proteins are these large macromolecular structures. And when you compare the two, a small molecule is really a little bit more to this size compared to a protein. And what's really awesome about these small molecules is that they will find a tiny little pocket in a protein or some other enzymatic target. They'll bind there, and they will have like great effects on biology. And so of course we use small molecules to, in this context, treat disease. And I would argue that natural products have had an incredible impact on human health. So there was a nice study that tried to summarize this. A few gentlemen from the National Cancer Institute, they studied the impact of natural products on clinical drugs. And between 1981 and 2014, it was estimated that about 65% of the drugs in the clinic either are natural products or they're inspired by natural products. And what that means is it'd be some kind of derivative. You make a slight change in structure or even a major change in structure, but at the end of the day, that structure is still based off of a natural product. The impact of these natural products on the antibacterial realm is even larger. About 3/4 of antibiotics in the clinic either are or are inspired by natural products. Now, you probably all know of one of the most famous examples, and not the first, but one of the first examples, and this was the discovery of penicillin. Anybody working in a hospital will know these as your beta-lactam class antibiotics. This is a wild story. I will not go into all of it, but just to say that there were, it's an amazing story that happened at the beginning of World War II. And it involves government, academia, industry. It involves, secrets and espionage, and it even has a part of the story that takes place right here in Peoria, Illinois. This is researcher Mary Hunt. She worked in the USDA lab, and she was the one who actually discovered the strain that produces large amounts of penicillin from this rotting cantaloupe she found in a Peoria market. And it was really this strain that allowed a couple decades to be added to the human lifespan. The bottom left is Dorothy Crowfoot Hodgkin, which her contribution was huge. She also settled a big debate about the structure of this antibiotic, penicillin. So this is a project that involved chemists, microbiologists, X-ray crystallographers. And the reason why I bring all that up is because if you just look at a list of, these are just a small representation of some of the drugs that are available both from plants and derived from microbes. And if you just scan over this list, I'm sure you're going to notice quite a few of them. And the point is, every single one of these drugs has a story like that. Maybe not quite as exciting, but they all have a story like that. And if you can imagine life without some of these drugs, it's quite difficult, and it would have a big impact on human health. For every single one of those small molecule natural products, they were all sourced in a very similar way. So what I've got up here is your typical antibiotic lead discovery pipeline, and it can be modified a little bit here and there depending on what your source is or how you set your discovery program up. But in general, this is how natural product drugs are discovered at the very front end of the pipeline. So in some way, you collect samples from the environment. So this pipeline is going to focus on microorganisms from aquatic environments. So we collect from lakes, rivers, or oceans. We then grow a large library of microorganisms, hopefully with little redundancy. Now, it's these microbes that produce the chemical weapons we're looking for. After we generate a library of microbes, we then try and generate a library of the compounds they produce. Of course, once we have this library, we then screen them against a bunch of different diseases or a single disease, whatever your program is interested in, and then you'll get a long list of hits. Once you have this list of hits, the next step is incredibly important. This is lead dereplication. This is basically, I don't want to spend the next 6 months to a year of my or my students' time re-isolating a known drug like rifampin or something. So we want to, in a very short time, hopefully under a week, be able to prioritize this list of hits from biological screening, whether it be a cancer assay or infectious disease assay. And we want to, using a tiny amount of material, figure out what is the likelihood that we're going to isolate a new compound or drug from this study. And then finally, lead identification, us figuring out what is the molecule that's causing all this bioactivity. And so now to transition into the real introduction for the project, what you see here is a picture of collaborators from Laos, Vietnam, and Illinois. And this is where my story starts. So like I mentioned before, this collaboration pre-exists my tenure at UIC. And I am particularly going to focus on two gentlemen, Dr. Dol Soeharto, who is at UIC, and Hung Nguyen, who was at the Vietnam Academy of Science and Technology, because they really started this collaboration. The partnering institutions involved are the Vietnam Academy of Science and Technology, which I'll refer to as VAST. There's the Institute of Traditional Medicine in Laos. And here at UIC, it was, well, what was formerly the Department of Medicinal Chemistry and Pharmacognosy and the Institute for Tuberculosis Research here. So all these partner institutions basically came together, and they, through funding from two different sources, the MacArthur Foundation for one, and for two, NIH International Cooperative Biodiversity Groups, they were able to accomplish a real lot within about 2 decades of research. And I'm going to sum that up in the next 2 slides. So what they were able to do, this ICBG grant was from the NIH was pretty awesome. It had 3 focal points: drug discovery, economic development, and biodiversity conservation. Now, in terms of this collaboration, they focused on a number of different diseases. It was cancer, malaria, HIV, and tuberculosis. So they were trying to discover drugs for each of those diseases, all the while trying to incentivize the conservation of biodiversity through economic development projects. These are very large grants with very large teams, and a lot of them were impactful. Now, I can give, of course, an entire talk on this, but I'm just going to summarize their accomplishments in 2 slides here. They had a number of different publications, patents, and books. There was a lot of student training, 4 PhD dissertations, but many scientists were not these 4 people. Many scientists were already in positions in Vietnam and at UIC and trained as a result of this program. And importantly, I think a number of different scientists were promoted to high levels of academia and government from this program, and were able to implement policy changes for economic development and biodiversity conservation. So they isolated a lot of different potential drug leads, and even developed a microloan program that raised 55 families above the poverty level. All right, so that is the backdrop from which I entered UIC. So I came in 2010, and Professor Soeharto, he's now a distinguished professor here, retired, but he was active when I was here, and he really took me under his wing and gave me some great advice. And all of those partnerships that you just saw were based off of medicinal plants or plants from the rainforest. So that entire grant focused on conservation in the Cuc Phuong National Park area, which is in the north of Vietnam and in Laos as well. And he came to me and said, my colleagues in Vietnam are really interested in starting a microbial drug discovery program based out of the ocean. They just formed a brand new institute, the Institute of Marine Biochemistry, and they're looking for advisors. And I said, oh, well, this is great. Let's go and do research. And so he gave me advice that was that I don't think I was ready to handle at that point. He said, all right, the next step is you need to get on a plane and shake hands. And of course, you know, I'm just starting a lab at this point. I'm super eager. I just want to get stuff done. I'm sitting on a startup package. So I've got some money to actually do experiments. And I'm thinking, oh, well, while I'm over there, why don't we, you know, collect samples, start to find new drug leads? And he stopped me. He said, no, go over and shake hands. And I didn't understand it then, but I understand it now. And what he meant was for international partnerships is you have to build trust. And so this was really the first thing that I was set out to do. So I went over there and did the handshaking tour as he suggested, and just you sit down and learn about each other, right? I have no idea what it's like to be a scientist in Vietnam, just as, you know, the scientists in Hanoi don't have a big idea of what it's like to be a scientist at UIC. And so we exchanged information, we broke bread together, and we discussed, and this is an important takeaway as well, we discussed what the goals of our partnership would be. And we came to a couple agreements. Number one, we were all passionate about training young scientists. So one of the first young scientists we trained was, this is Dr. Cuyen Vu, but at the time she was a graduate student, and the three of her mentors were Haidang, Huang, and Kuang. And so Kuang is going to be my co-PI in the remainder of these slides. So we wanted to train young scientists, but we also wanted to build this marine microbial library, and so and to transfer expertise to both students, postdocs, and to professors. And of course, we needed to obtain funding to support these goals. And to obtain funding, you need preliminary data. So I was able to, or we wrote a grant to get funding, about $50K from the Chancellor's Discovery Award at UIC, and the whole purpose of this was to just get preliminary funding so that you can obtain big grant money. What's considered big in the professor field is like, you know, usually greater than $1 million or something like that, right? That's just, I give that information just so the students can have an idea of what scale we're talking. So we took the money, and Professor Kung hired a couple people to do sample collection in the Cat Ba Peninsula. This is up Ha Long Bay in the north. And basically they sent the samples to our lab, and we quickly worked them up, grew microbes, and using an anti-cancer screen in the Burdett lab next door to me, we were able to publish a quick paper together to show that we have the capacity to transfer samples and work together. So this was part of our preliminary data. And the other part of our preliminary data was showing that we can readily travel between institutions and train people. So Kuyen stayed in my lab for 6 months, learned how to study microbiology, and brought those microbiological skills back, and she trained others as well. And it was at this time, now it's about 2015, so a couple of years have gone by, and now it's 2015, and I happened to stumble upon a really nice funding opportunity. This is an NIH D43 program called the Global Infectious Disease Research Training Program. This was absolutely perfect timing because we had just generated preliminary data, and we're looking to get major funding to carry out bigger ideas. And the purpose of the D43 grant is twofold. One is to strengthen the capacity of an LMIC. By LMIC, it's defined as low to middle income institution. So they want to strengthen the capacity of an LMIC to fight infectious disease and provide mentored training to students, postdocs, faculty in a health priority of that LMIC. So whatever disease disproportionately affects that country, that's what we would focus on. And so now this kind of forced me into learning a little bit about infectious diseases, okay? So one thing that we chose to focus on was the Mycobacterium tuberculosis. Obviously, this is the causative agent of TB. So globally, it's a huge threat. In 2019, there was about 1.2 million deaths, and nearly half of those were because of drug-resistant TB. And Vietnam has a disproportionately high burden of TB, and about 19,000 per year fall to the infection. Now, there have been a lot of partnerships between the two countries in the past couple of decades. Our own Centers for Disease Control has invested in clinical labs and surveillance systems to track TB, HIV, and AIDS statistics. And between 2004 to 2015, there was major investment through this program called the U.S. President's Emergency Plan for AIDS Relief. We invested over $700 million in medical infrastructure. And so what this led to, in my mind, was relatively strong clinical infrastructure. And so the way that I view drug discovery is kind of shown here on the top. So just from a basic scientist's point of view, I kind of view it as having three major components. You have basic research where your drug is discovered, translational research where a group of scientists take this drug lead and turn it into a viable drug candidate through several in vivo studies, mechanism of action studies, how to best deliver the drug. And then, of course, there's clinical research where you have your phase 1, 2, 3 studies and application and marketing of the drug. Now, through several discussions with our Vietnamese partners, we determined that there were some weak points that we were able to address here. And the bottom is specifically in regard to microbial or marine microbial antibiotic discovery. So although they have a relatively strong basic research program at VAST, it was not strong in terms of discovering marine microbes. And so we wanted to bolster this, and we wanted to link this with translational research, taking these leads and linking them with the relatively strong clinical infrastructure. And so the partnering institutions for the partnership that I was involved in was VAST and the NIHE, National Institute of Hygiene and Epidemiology, led by Dr. Dakain Dang. And then in the Pharmaceutical Sciences Department, there's myself, Professor Alessandro Eustachio, and at the ITR, Dr. Scott Franzblow and Sang-Kyen Cho. And so here are the specific aims of our proposal, right? We had three specific aims. One, we wanted to establish 3 independent investigators, basically professors at IMBC, and we were going to do this through long-term postdoc training. So the postdocs would come over to UIC for 2 years each, train in a laboratory of myself, Alessandra, and Scott and Sang, and then bring those skills back to Vietnam and implement them in their own independent discovery. Aim 2, we were going to provide both short and long-term training to 9 predoctoral students. And aim 3 is we wanted to build the research infrastructure and capacity through the creation of a series of assays to fight that combat TB, so we can readily discover leads. So it was a funny story about these 3 aims, and this is some more advice for younger scientists out there when you're applying for a grant. Always look at what that institute has previously funded, because that's going to give you an idea of where the space that you should try and be in. And I'm not saying to copy grants that are out there, but you don't want to propose something that's too far out of the funding range or funding scope of the institution. Now, fortunately for me, I did not take this advice, and I just happened to get lucky here. So when we applied for this grant, after the fact, after we had already received funding, I looked at what was previously funded, and these grants were all focusing on hundreds of trainees, right? Or 50 trainees here, 100 trainees there, and they do a workshop for 50 trainees and a workshop for 150, and then call it 200 trainees. Whereas our grant came and it was complete outlier and said, all right, we're going to have 3 trainees. But we made a good case for it, and we were luckily funded. We just came in with a different model. It was more of like a teach a person to fish model, right? So for our collaborators in Vietnam expressed interest in learning about how to grow microbes and do antibiotic discovery from them from the ocean. We were to just transfer that capacity over and then start a partnership. And what it would look like is this. We have a UIC mentor team that receives 2 postdoctoral trainees. And in turn, they learn and then bring the technology back after 2 years. And the same thing happens with 1 trainee from the NIHE, which is exchanged between the ITR and them. And between the 2, one side will provide screening for drug candidates and the other provides discovery of drug candidates. And the ultimate goal is to get this to be completely independent of UIC between 5 and 10 years. And if you look at this from an expertise perspective, what we bring to the table was chemistry, molecular biology, and microbial pathogenesis. And each trainee would spend 2 years in each of our labs learning these techniques and applying them to the drug discovery pipeline. So what I'm going to do in the next couple of slides is go over each of the aims. So for aim 1, I'm going to discuss each of the trainees. We had 3 fantastic trainees. The first one, the process was that Kuang would search through IMBC and VAST, find candidates, interview them, and then set up an interview with myself, and we would interview them. So it's a 2-stage interview process. And after this process, we were able to identify Dr. Lynn Nguyen. She was a fantastic scientist who had prior expertise in organic synthesis and natural product discovery, but from only plants. And so when she came to UIC, her job was to figure out how to build a microbial library and do antibiotic discovery from it. And what she would end up doing between her and Kuyen, who's pictured here in the back, what she would end up doing was building a preliminary library that was the foundation of what is now their overall institutional microbial library. So she was trained in microbiology, the operation of pretty intense bioinformatic pipelines, a lot of which are freely available, the identification of bacteria, project management. These last 3 are really important because we're not just training them to do science, we're training them to be PIs or professors. And that, again, there is no handbook that I know of that covers how to do this job. You kind of just learn on the fly. And so we taught politicking, how to write grants, how to manage a team. And I would let her into the office for her to see, me banging my head off on my desk, or I wanted her to see me fail. And I wanted her to see every time that a grant got rejected and every time I had absolutely no idea what to do. I wanted her to see all of that. And she did. And I think she's applying some of those lessons today, learning from all of my failures. So she ended up co-authoring 2 studies, and I mentioned that she curated this library. This library was made off of several different trips to Vietnam. So this is the team collecting. We would go out on boats, but there was a giant tropical storm heading to an island. We were on a military island, Phu Quy, and so we had to stay inland before the storm hit. And then usually what collections look like is this bottom one. An interesting story about this bottom one. We had to deep dive to find the shipwreck, but we dove down, not necessarily because we were looking for the shipwreck, but because people started dynamite fishing next to us. And so it's exactly what it sounds like. So we had to kind of dive down deep to escape that. That was a pretty scary story. So she collected these samples, and then the team goes back, and wherever we are, whatever hotel we're at, or wherever we can find resources, we set up shop and start to grow these bacteria, build the library. And then Lynn used a bunch of different bioinformatic platforms to create this library, transferred it back to IMBC, where it now sits today. I love bragging about my colleagues here because in the past 5 years, this is not a list of all of our publications. This is a list of the publications that are completely independent of UIC. So there's over 10 publications here where professors have just learned the techniques and have, because the talent pool is so great at VAST, they were able to basically, once they learned these, they were able to just start pumping out their own independent studies, which is pretty awesome to see. So they no longer really need us to do microbial isolation and discovery. All right, so for the second trainee, Dr. Tuan Tran, he joined the laboratory of Dr. Alessandra Eustachio. Now, he came in also with a background in organic synthesis and plant natural product discovery. And what he learned was training in biosynthesis of natural products or antibiotics. This is how bacteria use building blocks to synthesize and make these molecules, and a very hot area of genome mining. So genome mining is a really a novel way to try and find new drug leads. And I think some of the major impacts that Tonine has had have been the development of coursework while he was here. So he worked with Alessandra to teach biosynthesis, natural product biosynthesis, and what is, to our knowledge, and we really tried to find other programs like this, but this is the first research program in Vietnam to introduce this topic. And so Tonine plays a really important role here in that he's going to teach the first generation of students to really dig into this topic, which is pretty awesome and inspiring. And the third trainee, Dr. Lian Pham, she's from NIHE. She came in with expertise in medical molecular biology, and her training at the ITR was in a whole battery of in vitro TB assays. She also worked to try and determine the mechanism of action of antibiotics, because of course we're interested in how they work as well. We want to know if they have a novel target. And she studied project management and grant writing as well. She was co-authoring a study while she was here. She performed the screening for these nanomolar TB inhibitors. And then she has since returned back to NIHE, where she's currently setting the lab up. I'd say we're about 70% there. There's still a couple major pieces of equipment that we're working on acquiring. It's just, there's a lot of red tape in that particular process. So it's taken longer than we would have liked. But the good news is that she's already started doing in vitro screening with IMBC, which is right down the street from her. All right, on to the second aim. This is just training of 9 pre-doctoral researchers from IMBC. So the way that we did this was we designed a bunch of short courses that were tailored to antibiotic discovery for infectious diseases. We gave access to both graduate and undergraduate course material and recordings. The D43 grant provided funds to allow a lot of the pre-doctoral students to present their scientific research at regional and local meetings. For young scientists anywhere in the world, it's important for them to go to conferences. I mean, that's where I learned how to, that's basically where I got interested in doing what I'm currently doing today. So going and presenting your work connects you with a larger network of researchers. And one of the funnest parts of the training also, as you see in the picture on the top left, was hands-on field expedition and training. The team collectively over the course of a couple of years, I would go back once a year to do a 1-week expedition. And then 1 week I'd stay and just talk with my collaborators and we'd plan ahead. But we collected over 200 samples from 20 different collection sites, and these came from about 2,000 nutrient plates. Here's a map of Vietnam on the left. And each of the yellow pins is a place that we collected. So we're able to go to a lot of different sites. We were very fortunate, and Cuyen in the middle was fantastic. She did a lot of the planning for these expeditions. My job was basically go into the water and dive with one of the local dive partners, this is Mr. Fung. And then the team of postdocs and undergraduates would then work up the samples and build the library when we were done. So it was a true collaborative event. And this is just pictures of the team processing samples in everywhere from houses to hotel rooms to vessels. And they were phenomenal, absolutely phenomenal. And the final picture of us loading up a boat, which in my opinion was a little bit overloaded. It was quite scary. And finally, to aim 3, the purpose of aim 3 was, again, enhancing biological assay research infrastructure at NIHE. Some of the assays that Lian learned here were an assay to target replicating TB, which is MABA, or also an assay that was developed in the ITR to target dormant or non-replicating TB LoRa. She learned a suite of cytotoxicity assays, of course, because when you're developing an antibiotic, you don't want it to be toxic to your host, so you want to minimize cytotoxicity. And then she learned a number of other different assays that I am absolutely not qualified to talk about as well. And so currently, as I mentioned before, she's working on obtaining both pathogenic strains of TB and of obtaining equipment to help her complete her screening. But she does have some of the basic MABA assays underway, and they already are screening samples for IMBC, which is pretty cool. All right, so in this 5-year period, some of the accomplishments of our team are we instituted 3 early career investigators, and they have just been crushing it. This is unbelievable. Part of the training of the postdocs was, I said, all right, by the end of year 2 here, we're going to have you write and submit your own grant. And we actually saw an opportunity from the NIH that was specifically tailored to foreign investigators to fight infectious diseases. And we said, oh, this is amazing. Submit this grant to the NIH, and it's an R01 grant, so it's big money. Submit this, and it'll be great practice. You'll get feedback on it, and maybe you can turn this into a grant when you go back to Vietnam. Well, they actually submitted it, and they got the grant, which is crazy. Meanwhile, I'm getting rejected left and right from the NIH. They submit their first grant on their first try, and they're funded. So they are currently, they basically go back to Vietnam as rock stars and have an awesome R01 grant, where all 3 trainees are involved. 2 are the PIs and 1 is a co-I. Dr. Tran has acquired additional funding as well, and he's already mentoring his first graduate students. We've set up, again, 70% of the way there, set up an in vitro screening program. They've published several times without UIC as collaborators. 5 IMBC faculty within the Institute, this is interesting, I thought, have chosen to focus their entire programs, or at least part of their program, on microbial natural product discovery. That explains the level of publications that we're seeing. And what I think is most important, and we had this in the back of our minds the whole time, was we wanted to establish institutional knowledge. And this, I think, is what set our grant apart from other grants that were funded. This, we really wanted to focus on teaching the teachers. And hopefully that's what we accomplished. And we'll really see how well that works out in the next 5 years, 5 to 10 years or so. So to move on to the final chapter of future plans to enhance disease fighting capacity, this is where we see the collaboration going. We've had a lot of talks as to where we see this going. And it usually starts by me telling my collaborators, I don't know anything. So where do you find weaknesses, and where do you think we can synergize with one another, and how can we bolster infrastructure at IMBC? That's usually where the conversation starts. So just a reminder, this is where the program was, and this is kind of where we're at right now. IMBC now collaborates with the Institute of Marine and Environmental Resources in Haiphong. This is just southeast of Hanoi. And then they also collaborate with the Institute of Oceanography, which is in Nha Trang, which is kind of in the south of Vietnam. And NIHE is screening samples from IMBC for all of this. Now, I think my long-term goal for this project is really this. And so we've done a lot of we've had a lot of discussions, and this is not for the next 5 years, okay? Because currently we have a submission in that's currently under review. The grant should be reviewed in the next month or so. This is phase 3 right here. So phase 2 is what we just submitted, and that will take us up until 2025 or so, or 2025, 2026. This is phase 3. We would like to create a screening center for early stages in drug discovery, starting at like basic research that spreads throughout Southeast Asia. Like we're starting to line up collaborations in Singapore, Malaysia, the Philippines, Thailand, and we hope to build some preliminary data over the next 5 years. And then in 5 years, we'll submit another renewal that focuses on really making this a regional screening center. And this is the grants that we just submitted. So this is kind of phase 2 before what I just showed you. We have 3 areas that we're focusing on for the next 5 years: undergrad and predoctoral training through course development, more long-term postdoc training, and professional training. So just to dig into each of these a little bit, for the first aim, we want to develop what we consider to be relatively comprehensive drug discovery coursework that has a focus on infectious diseases. And so the whole point of developing coursework, of course, is to educate young scientists, but it's also to try and recruit the next generation of researchers that are going to try and combat infectious diseases. So giving them a strong foundation early on in their scientific careers. The second aim is going to be to develop, this is the long-term postdoc training. We're going to be taking 2 students training back at UIC again, and they're going to focus on bioinformatics, which is just basically using computer programs to process data. It sounds simple, but it's not. It's very easy to collect large-scale data. It's not easy to analyze large-scale data and visualize it and communicate it in a way that's effective. So we'll focus on bioinformatics and human microbiome research. We just hired a new professor, Professor Matt Henke in our department, who is in a great place to be able to expand the repertoire of IMBC into this field. And finally, aim 3 is going to be to train and support leadership. What I mean by this is, when we were sitting and discussing what some of the weaknesses were at IMBC, one of them was administration. And the professors, the professors do a lot more than we do here. So the professors there not only have to do all the science, but they have to do all the administrative work for their grants. I have an entire team that deals with like compiling the grant, submitting it, and dealing with the NIH and helping me manage the budget. They do everything. And so what we're hoping to do is train staff to work at IMBC to kind of lighten their load and free their time up so that they can focus more of their energy on biomedical research. Okay, and with that, there's a lot of people to thank. I'm the one who gets to sit up and brag about my collaborators, but they do most of the work. So for mentors at UIC, Scott, Sang, Alessandra have been awesome. Elizabeth Ryan is somebody I did mention, but she is an administrator here, a grants administrator, and without her, none of this would have been possible. I will say this is it is so complicated to move money between 2 countries, particularly when the time difference is 12 hours. So, noon here is midnight there, and she has just absolutely been indispensable. Professor Soharto has just been a constant source of inspiration, and he really, in addition to Professor Hung, they started this whole partnership. And here they are. Here's Hung, Professor Soharto, and then my partner in crime, Professor Kong, and Hong, who is right over here. So these are all co-PIs. Kong was the PI in the first the first section when we started this grant, and Hong is now taking over as the PI because Kong just got promoted. So we've had amazing collaborators there. The trainees, Lian, Tonain, and Lin have been outstanding. And then the NIH has been very supportive of our work. This is through the National Institute of Allergy and Infectious Diseases and the Fogarty International Center. Yeah, so with that, I would be happy to answer any questions that you have. And thanks for listening to me. I really appreciate it.
Thank you, Dr. Murphy. That was great.
Thank you, Dr. Murphy. It was wonderful to hear about how your global health partnership was formed and maintained and the directions that it went. It was fascinating to hear about the success stories, seeing the independent researchers obtain publications and large amounts of grant money. So that's truly a success story and a lesson for all of us. We welcome the audience to submit questions. Please simply raise your hand and we will ask you to unmute yourself. And don't be shy. We do have a question from Carla. Jeannie, if you could please have Carla unmute.
Hello. Hi, thank you so much for this lovely presentation. I actually am a graduate from the School of Public Health as well, currently working as a family medicine resident and thinking about doing some projects in Guinea-Bissau, West Africa, where I'm from. And I just started linking and connecting with some local organizations, Ministry of Health. And I wanted to know what are some of the struggles that you faced in establishing such a long and strong partnership, and how did you overcome those struggles?
That's a fantastic question. And the answer to this question will be the key to your success. I do not have all the answers, but here are some that I have. Some struggles were communication, right? For instance, my Vietnamese is not very good. I've tried and I'm still trying, and and they bear with me and they're very patient with me. But of course, in the sciences, I am very fortunate that I'm from the States, because at the current moment in time, English is a very common language throughout the world in the sciences. It's obviously not the only language, but so because of their grace, we are able to communicate. But communication is still very difficult, especially the time differences. I'm used to working with a lot of collaborators at UIC where I send an email and it's answered within an hour. And so I said, oh, I have an experiment that I'm going to do, or there's something that I want to get done. But in terms of having collaborators overseas, where you have incredibly different both cultures and time differences, the key is planning ahead. You really have to plan ahead. Sit down, both of you, hopefully in person. Over Zoom is okay too. In person is way better, and make a schedule and just say, hey, here, like for instance, we tried to write a grant, right? And we said, okay, here's the deadline. And then you work backwards from the deadline. Like, all right, we want to have the grant completely done 2 weeks beforehand. Well, what do we need to do that? And you list out the things you need to do, and you put specific deadlines. And that's one way to avoid miscommunication over long distances, because you're going to send an email and you might not hear a response for a day or even a couple days. And depending on the person, because everybody has different work ethics or different holidays. I mean, there's the Tet holiday that I have to be aware of in Vietnam. And so there's a lot of things that end up delaying what you think is a perfect schedule. That is the major thing, I would say.
Thank you so much.
Yeah, no
And
problem.
my second question, it has to do with this model. I think it's really beautiful how it's like almost train the trainer, and how sustainable, like, so are they, well, two questions. First, how did you select the scholars, or, how did you know they would actually go back? Because I think that that's some of the challenges that some countries face, that they leave abroad and then they never return. That's one. The second question is in terms of sustainability. So now, do they only write their own grants and they're able to get them right away, or are there other sources of funding that you've created?
Yeah, for the last question, a little bit of both. There are institutional grants that they're lucky, and every country is totally different on where scientific funds come from. And that is something that is, I am totally ignorant of. Like, I've done my best to learn, but your best source is going to be your partner. And so, I guess one way that our model differs is that when I started this collaboration, we went in as like 50-50 partners. And I guess from talking to the NIH, a lot of grants in the D43 sector don't really start as that. It starts out with the PI in the States. And by like the second or third round, when they're refunded, they hope to train somebody to be a PI. But we were just fortunate to I have great partners there that are awesome basic scientists. And so immediately start a 50-50 partnership. And so to kind of answer one of your questions, I rely on them to weed the candidates out, right? And you have to, they know their institution better than I ever could. And so they, and what you can do is you stress, listen, here's the parameters of the trainee that I think would be ideal. And then you ask them, do you have anything to add? And say that one of these parameters is that they have to return to the home country, or else this is not going to be successful. And you really stress that. And at every stage throughout the interview, that is emphasized at every single stage. And then even when the trainees come, at every stage, everything you do is built to have their career be back home. Because if you built their career to be in the States, they're going to stay. Right? I mean, or they're not, they might not stay, but I mean, because if they build an infrastructure that is here, then you kind of tie them to that infrastructure. So it's important to build an infrastructure that is back in the home country.
So while they were in the US, did they go back as well, like for, few periods of time, just to, apply what they learned?
Yeah, every year we would go back for different amounts of time, and I would go back for a couple of weeks, and yeah.
Thank you so much.
Other questions? Alexander, please go ahead.
Yes, thank you. Hi, Brian. It's wonderful presentation and a great story. I have sort of a drug discovery question. So the different human diseases as well as animal diseases are probably more prevalent in specific areas on the planet. So do you think there's a high chance of finding the antibiotic for a specific disease if you look at that place where this disease is more prevalent, or it's totally random and doesn't really matter?
I'm not going to take the easy way out of here, but I think both. So I think there are strong ecological reasons to believe that you're more likely to find a drug, for resistant bacteria in an environment where resistant bacteria exist, right? But that isn't to say that you can't find a drug for a resistant bacterium in a completely random place. I mean, we found awesome antibiotics at the base of a hydrothermal vent, in 100 feet of water in ice, in freezing cold waters of Iceland, right? So there's not a ton of human relevant diseases there, but we're really encouraging scientists in our fields to take a more ecological approach to it and consider where is the disease or bacteria like the disease, and maybe search there for antibiotics.
So then, following the same logic, would it be reasonable to search for proper antibiotic producers in the hospital environment where they encounter a lot of resistant bacteria?
Yes and no. I don't want to. I would lean a little bit more toward no, because it's an environment where what we've relied on is diversity. So the more it's kind of like an attrition-based approach to drug discovery. The more microbes we can cultivate, the better shot we have at discovering something new. In hospitals, you find a lot of pathogens in hospitals, but there aren't a lot of like actinomycetes or firmicutes or other bacteria that might harbor weapons here. But what I think would be interesting is looking at other pathogens and their ability to produce small molecule factors. But to one up you there, we don't go to hospitals. One thing we've tried in recent years is we go to wastewater treatment plants, and we sample from multiple stages within a wastewater treatment plant.
Thank you very much.
Yeah.
Ursa, please go ahead.
Hi, I am Tirsa Ferrer. I am from the Division of Pulmonary and Critical Care Medicine. Thank you for this presentation. It was it was awesome. Thank you so much.
Thank you.
My question is more related to your overall experience, combining doing the research, the mentoring, but also going to the field and doing the scuba diving, which I think is amazing. But when you mentioned the dynamite fishing, there's also some, safety concerns there. How you do the groundwork to establish safeguards so you and the team are safe and can complete the expedition safe?
Awesome question. We rely heavily on local knowledge. I always humble myself and walk into an area. I never try to hide behind my dive certifications, right? I think a lot of humans do this. We're afraid to show our faults, right? Because we're insecure. I'm very insecure. Everybody is insecure, whether we admit it or not, right? And so for me, I don't, I'm a person who walk into an environment and be like, I don't know anything about what we are about to do. And I pretend like I'm a beginning diver. We'll team up with local dive shops and local experts within the area to understand, what kind of research is happening in this area. Like, is there a water treatment plant, or is there an agricultural runoff near the place we're diving, right? I've gone down in sites before where there's just sunken fishermen's nets all over the place. And this was not ideal because you can easily get tangled in these nets. But you just rely on local knowledge and local dive shops and consult more than one of them. And then you compare answers, and then you go out and essentially you make the call when you're out there. If you don't think it's safe, you don't go. And I have no problem canceling a dive or a, but yeah, it's a complicated one. Good question.
Thank you. Thank you.
Hey, Nancy, thank you. Please go ahead.
Hi, Brian. Wonderful talk. I just wanted to ask a little bit about something you alluded to already in terms of your culturing methods. So the more you can culture, the more you can discover. Do you have work on the pipeline to expand the culturing as well, or how do you approach that?
Oh, how much time do you have? I don't know. It's funny because having a background in organic chemistry and being trained in microbiology for a short time and then doing it for about a decade, I still feel like I know nothing. And I still feel like a lot of it from my end has been 50% planned and 50% complete luck. To access new microbes, we have always gone to unique places, and that is definitely successful, right? Hydrothermal vents in Iceland, or even, you don't even have to dive off it. We can go into terrestrial environments which have like basically bubbling pots of minerals, really unique microbes there, hopefully really unique antibiotics. What the IMBC is doing is up to them. One of the areas that they're starting to get into now, which is awesome. We haven't gotten into it quite yet. We've done a little bit of it. They're starting to collaborate with people who study caves. So they're going, because there's an incredible system of caves across middle to southern Vietnam, and they're collaborating with some people from the States and some not from the States, some from southern Vietnam, and they're collecting cave bacteria. And the culturing methods, there's some known about culturing cave bacteria. There's some publications out there, but from a very non-expert point of view, like a lot of that to me is guesswork, but I'm sure a microbiologist would slap me in the face if they heard me say that. So
Thanks, Brian.
yeah, no problem.
We do have about 10 minutes longer if there are more questions from the audience. I'm not seeing any more people come forward. So it looks like we will give folks back some time in their day. Dr. Murphy, thank you very much for joining us today as a presenter at the UIC Global Health Colloquium. We do hope that folks tune in next month, December 7th, for LaRon Nelson from the Yale School of Nursing. All right, thank you everybody for joining. Have a wonderful day. Oh, of note, this will be posted on the website at the College of Medicine Center for Global Health. So if you'd like to reference it again or share it with others, please do so. All right, thank you.
Thank you, Dr. Murphy. That was
Thank you.
great.
Thank you. I appreciate the invite.
Thanks.